Pop quiz. What happens when a cell's internal timer breaks? A. It dies. B. It rests. Or C. It divides uncontrollably. Lock in your answer. The correct answer is C. When cell cycle controls fail, like checkpoints or CDK breaks, cells keep dividing, which is how cancers start. In this video, you'll learn how the cell cycle is regulated, what the key phases are, how cyclins and CDKs control each step, and what happens when regulation fails. The cell cycle is the ordered series of events that prepare a cell to divide. It has 4 phases: first gap or G1, synthesis or S, second gap or G2, and M phase, which includes mitosis and cytokinesis. Let's walk through them. In G1, the cell grows and builds materials for later. During S, DNA is replicated. Next, in G2, it produces the proteins needed for division. Finally, in M phase, the nucleus divides and the cell splits. To move from one stage to the next, cells rely on cyclins, proteins whose levels rise and fall in a repeating cycle. These cyclins then activate enzymes called cyclin-dependent kinases, or CDKs. When a cyclin binds a CDK, the pair forms an active complex that drives the cell forward, as serine-threonine kinases. These complexes add phosphate groups to target proteins, turning parts of the cell cycle machinery on or off at precise moments. To keep the timing specific, each phase uses distinct cyclin CDK pairs. For example, cyclin D with CDK4 and 6 promotes G1 progression. Cyclin E with CDK2 helps trigger DNA replication in S. Cyclin A with CDK2 carries the cell through S into G2, and cyclin B with CDK1 controls mitosis in M. In addition, checkpoints between G1 and S and between G2 and M act like quality control gates, ensuring the DNA is intact and conditions are right before the cell proceeds. So far, we have outlined the phases, seen how cyclins and CDKs push the cycle forward, and noted the checkpoints that verify readiness. Want to check your progress? Tap the link for a quick practice test and a concise study guide tailored to this lesson. Quiz yourself, skim the highlights, and jump back in. Now, regulation works in both directions. Activation moves the cycle forward, and inhibition applies the brakes. Cells pause progression using CDK inhibitors or inhibitory phosphorylation when repairs or resources are needed. In humans, proteins like P27 bind to cyclin CDK complexes and block their kinase activity, preventing premature division. In late G1, the cell also faces a restriction point, a commit or wait decision that checks for growth signals, nutrients, and DNA integrity before it proceeds. When DNA damage or replication errors occur, checkpoint proteins stop the cycle for repair. If the problem cannot be fixed, they trigger apoptosis. This control ensures that only healthy, fully prepared cells divide, preserving tissue integrity. By contrast, when checkpoints or inhibitors fail, or when cyclins are overproduced or CDKs remain persistently active, the cell loses control of the cycle and proliferates unchecked, while errors accumulate, a hallmark of cancer. In response, many modern therapies aim to restore control by inhibiting CDKs and reinforcing checkpoint signaling. To recap, cyclins rise and fall, CDKs drive each phase, and checkpoints pause for repair. When this regulation fails, cells divide uncontrollably, leading to cancer, insight that guides today's CDK-targeted treatments. Now that you've watched the video, let's see how much you remember. Head to the link in the description where you'll find Quizlet content made just for you.